Happy New Year, everyone. This is Brain Ponderings. I'm the host, Mark Matson. Beginning next week, we'll get back on the regular schedule with me talking to guests, typically neuroscientists, neurologists, psychologists, so forth, uh, about their particular area of expertise in research. Today, I'm going to continue with this very short series of presentations on problems with the United States healthcare industry, and I'm going to focus on the pharmaceutical industry in particular. Last presentation, I talked about the food industry. Um, and this slide, this initial slide, by the way, if you're not watching on YouTube, I'm showing slides. And also, I should say, if you like these podcasts, do subscribe to the channel. So this diagram I showed before, which are at the center in red, are some of the major areas that of concern in terms of our poor healthcare system, in that we spend more on healthcare than most other industrialized countries and that still have poor health outcomes overall. And there's a lot of different issues here. Today, again, I'm going to focus on the pharmaceutical industry, some of the problems with it, and then at the end, I'll talk about some potential solutions, or at least interventions that might help with the problems I'm going to talk about. Okay, so most of you know this. In the United States, we spend, as I said, more on healthcare than, for example, Canada, most European countries, definitely Asian countries, and we have poor health outcomes in terms of lifespan, chronic diseases like diabetes, obesity, cardiovascular disease, even many types of cancers. And so in this next slide, I'm going to summarize what I'm going to talk about today in terms of the problems. Drug costs. So you've heard in the news, if you're in the United States, certainly, that the drug prices are much higher than other countries. You may have heard about problems with insulin, for example, or EpiPens for people who have allergies to bees or other types of allergies. And so if you compare just overall, on average, the price of any particular medicine is at least three times as much in the United States as it is in, for example, Sweden, Canada, Germany, uh, Asian countries, Japan, South Korea, for example. Um, and for each individual, on average, an individual spends $2,000 a year on drugs. Now, if they're covered by health insurance, that will probably pay for most of their drug costs. However, there's still about 25% of people in the United States who don't have health insurance. There's been a increasing rate of drug costs, and it's much more than the rate of inflation, 5 to 10 times. Drug companies often justify the prices they charge for drugs by saying it costs them a lot of money to develop the drugs. But it turns out that analyses have shown that they're actually overstating by 4 times how much they actually spent on developing drugs. Then another issue around this is that a lot of the basic research, or in fact, most all of it, that drug companies use to develop their drugs, for example, identifying a particular signaling pathway that affects the growth of cancer cells, or a particular neurotransmitter or other type of signaling system in the brain that affects plasticity or mood or vulnerability of neurons to disease. And the the pharmaceutical companies then use uh, that information on to make decisions on what targets, what molecular targets they're going to focus on, and then they may develop a screen, or they may actually use screens developed through NIH funding. Okay, another issue with the pharmaceutical industry is that they game the system. Uh, for example, there's patents last only so long when they develop a drug, 20 some years typically. And so what happens is a company may develop a drug, and then by the time it gets FDA approved, maybe it's only on the market 10 years, sometimes even less. And so then they make another drug that does the same thing as the initial drug, and then they um, market it as being better. And I'll talk about why that's a problem. Namely, they haven't established that it's better in clinical trials. So examples are commonly used antidepressants. Many different companies have developed drugs that do the same thing. For example, serotonin selective reuptake inhibitors, SSRIs, or serotonin norepinephrine reuptake inhibitors, SNRIs. And even in the case of what are called biological, so essentially, for example, antibodies or um, proteins, peptides, such as GLP-1 receptor agonists. Um, and again, the basic research showing that that GLP-1 uh, counteracts obesity and diabetes was done basic research labs at NIH or at universities, and then the drug companies, they take GLP-1, which is a small peptide, 30 some amino acids, and they change the amino acid sequence a little bit, or they otherwise modify that peptide and find that it has the same effect as GLP-1, and then they market that as whatever, Byetta, Ozempic, Wegovy, etc. Okay, so essentially copycat. Uh, and then another big problem is the drug companies actually spend more, nearly twice as much, on marketing their drugs as they do on actually developing them. And we're one of the very few, in fact, there's only two countries in the world that allow direct-to-consumer marketing. You see ads if you're on, uh, well, if you're on the internet, many outlets, if all the news outlets, you'll see advertisements for drugs, and they kind of glorify these drugs as being cures or having dramatic effects when oftentimes they have only modest effects and a lot of side effects. Um, so the ads generally overstate the benefit to risk profile of the drugs. Another big problem is lobbying. Drug company representatives, they lobby Congress in the United States, uh, senators, congressmen. They often, these drug company representatives, some of them will actually physically go to doctors' offices and offer free samples of drugs, and sometimes even perks. Um, for example, I was in one doctor's office for some orthopedic problems I have, and they had one of the reflex hammers you use to test somebody's reflexes, and it was from uh, a drug company had given it to the doctor. Uh, Many conflicts of interest, um, particularly around clinical trials, where many of the clinical investigators involved in clinical trials receive money or other perks from the pharmaceutical company from which the drug was developed and being tested. And I also talk, there's some other issues around transparency in the data availability from some of these clinical trials. And then lastly here, actually pharmacy companies, this isn't well known, but they often have managers that skim off dollars off the uh, by doing price markups. And there's a nice, very concise article on these problems with the pharmaceutical industry by Hoffer in 2019 in the American Journal of Medicine. That references here on the slide. If you want to go to that, you can find that on PubMed. Okay, here's just a few examples of drug price gouging. Insulin. Um, this diagram at the left there, the size of the circle represents the relative price of insulin of the different countries. So the United States is like manyfold, like 5 times as high as most other uh, industrialized countries. And there's been there was a dramatic increase in the price of insulin between, well, approximately 2012 and 2020 or so. And then very recently in the last year, uh, Congress and the president enacted legislation that greatly lowered the price of insulin. It capped it at a certain low, reasonable level. Those other examples show here some anti-clotting drug, Liquius, and EpiPen I mentioned, statins, which are used to lower cholesterol levels, and sitagliptin, which is a diabetes drug. And this just shows the relative prices in the United States versus Canada. And you see that about 4 times as much in the United States as Canada for the same exact drug. Now, again, if you have insurance and you're fortunate to have good insurance, your insurance will cover some of this. But nevertheless, that this is price gouging. That money's going oftentimes to not necessarily to research and development at the drug companies, but to marketing and to paying their top executives a lot of money. Okay, um, spending on on medicine and medical intervention, so healthcare, it's a major problem because a major cause of bankruptcy in the United States is medical debt. Okay, and this in this slide it shows results of a survey showing how greatly impacted the lives of people are by not being able to pay medical expenses or having to cut back on basic necessities in order to pay the medical bills. Okay. Now, in the next few slides, I'm going to show these tables, and I cite the reference here in Experimental Biology and Medicine 2019 of this article. It lists many of the drugs used for various cancers. Of course, cancers are devastating diseases, and we want to have effective treatments. There are effective treatments. Some of them more effective than others. There's a lot of excellent work going on in coming up with new ways to, for example, stimulate the immune system to attack cancer cells. And that's all good. Uh, but the cost of some of these drugs are, whether they're a small chemical or an antibody or some other uh, factor that's affecting the immune system, Some of them are extremely expensive, and this is shown here, the treatment costs here in the right, $10,000, $20,000, even $100,000 for treatments. More examples here. Uh, Very expensive. Okay, and then the efficacy of the drugs used for cancers, as I mentioned, is highly variable. Some of them can have dramatic beneficial effects, others not so much. And the cost of the drug is not, of any individual drug, is not directly related to how effective it is. Okay. This is a neuroscience podcast focusing on brain research and translational research, neurological disorders. So I'm going to talk for the most of the rest of the talk about brain disorders. I'm going to spend some time on Alzheimer's disease. It's a big problem. Currently, there's about 7 million Americans living with Alzheimer's disease. Uh, 1 in 3 older adults dies with dementia. In other words, if you live to be 65 or older like I am, you're likely to die with dementia, either as a cause or with dementia, but with another immediate cause of death, like a heart attack, stroke, etc. It's a huge burden on families and on the healthcare system. It's estimated that in 2024, cost the nation $360 billion by 2050, 25 years from now. It will cost $1 trillion for this includes loss of productivity of the affected individual, the cost on the family, uh, costs for caregivers, going into uh, long-term care facilities, nursing homes, and so forth. Okay, as you know, if you look at the brain of someone who died with Alzheimer's disease, there's two dramatic features. One is the accumulation of amyloid plaques, extracellular accumulations of these aggregates of this 40 to 42 amino acid amyloid beta peptide. So that's the amyloid plaques. And then inside of neurons that are degenerating and die is the accumulation of a protein called tau, TAU. It's a microtubule associated protein. So it's normally plays a role in controlling the cytoskeleton, the structure of the neurons. And in Alzheimer's disease, this tau becomes hyperphosphorylated and it falls off the microtubules, uh, and the microtubules depolymerize. And this is probably contributing to their cell death, certainly contributing to their structural issues. Okay, the amyloid beta peptide, A beta, which is shown in red in this slide, is a small, as I mentioned, 40 to 42 amino acid segment of a much larger amyloid precursor protein, or APP. This APP is a transmembrane protein, so it's in the outer membrane, plasma membrane of neurons in particular, but other cell types too. And so how does this amyloid peptide end up getting liberated from its precursor protein and then aggregating? Well, there's two enzymes that cleave the amyloid precursor protein to liberate A beta. One is called beta secretase. It cleaves at the N terminus, amino acid terminus of A beta. And the other is called gamma secretase, and it cleaves at the C terminus of A beta. So then you get A beta monomer released, and this can actually be removed uh, from the brain and enter the blood, actually. So normally in healthy people, this A beta does not accumulate, but with aging and Alzheimer's disease, there's changes that occur in the brain. For example, oxidative stress, inflammation, maybe, that promote self-aggregation of the amyloid beta peptide. Okay, then there's also an enzyme called alpha secretase that cleaves in the middle of the amyloid beta peptide sequence. It releases what we call secreted APP alpha from the cell. And we have shown, and others have evidence for uh, uh, this too, that this secreted APP alpha has a normal physiological function or functions. I'll briefly talk about that later. Okay, so how does this amyloid beta peptide does it? And if so, how does it damage neurons? Well, these illustrations here in this slide that I'm showing are from an article I wrote for Nature uh, a little over 20 years ago. And unfortunately, there hasn't been tremendous advances, and I'll tell you why. Well, there's been quite a bit of advances in basic research, but not so much in translation. Okay, we showed when I was at University of Kentucky, a professor there in the 1990s, beginning in the early 1990s, together with Alan Butterfield and the late Bill Marksberry, that this amyloid beta peptide, when it's in a self-aggregating form under oxidative conditions, that is, in the presence of oxygen and maybe low amounts of free iron or copper, it aggregates on the membrane of neurons and cause what's called lipid peroxidation. So free radicals are generated that attack the membrane lipids. And we have shown through a series of studies, some published here, that through this mechanism, the amyloid beta peptide, by inducing oxidative stress, impairs the ability of neurons to take up glucose, and it impairs the ability of the neurons to control the electrical potential across their membrane. And it does that by impairing what are called the sodium transporter and the calcium transporter. Okay, and this we showed renders neurons vulnerable to excitotoxicity. And I'll talk about that. We've talked about excitotoxicity in other episodes of brain ponderings. And I should mention, we now have I've now done over 100 episodes, well over 100 episodes of brain ponderings going back, I think about 2.5 years, maybe 3 years. And so if you haven't looked back at some of these early podcasts, you might want to, because uh, they cover a lot of interesting areas of neuroscience with top people in the field, including Nobel Prize laureates, National Academy of Science members, etc. And I do try to get the top people in the field, and usually do, because I'm a neuroscientist and know a lot of them. Okay, so I mentioned excitotoxicity. So what's that? So glutamate, the excitatory neurotransmitter in the brain's most important neurotransmitter, as I talk about extensively in my book, Sculptor and Destroyer. Okay, when a neuron fires, glutamate's released from a presynaptic terminal. It binds to receptors on the postsynaptic membrane. A certain type of those receptors, called AMPA receptors, are sodium channels. So remember, sodium is high concentration outside neuron, low inside. It has a positive charge, so you get, when this AMPA receptor channel opens, you get sodium influx. That then depolarizes the membrane, and that, together with glutamate binding to another type of glutamate receptor, the NMDA receptor, causes that receptor channel to open, and it's a calcium channel. So bottom line is when synapses are activated by glutamate, it causes sodium and calcium influx into the postsynaptic cell. Now, normally, this sodium and calcium is rapidly removed by uh, membrane, energy-dependent membrane transporters in the membrane, but the amyloid beta peptide can impair the ability of this neuron to restore its membrane potential, and that we think contributes to an excitotoxic type of death where you get sustained elevation of intracellular calcium levels that can lead to, for example, depolymerization of microtubules and damage, even death of neurons, which is easy to demonstrate. Uh, in cultured neurons, you expose them to high levels of glutamate, it kills them. You expose rats or mice to, or even humans by accident, to certain excitotoxins that activate glutamate receptors. It kills neurons. So again, I talk about in this my book, Sculptor and Destroyer. Now, the only drug that has been shown in clinical trials to slow down significantly the the progression of Alzheimer's disease, that is the neurodegenerative process, is a drug called memantine. And in this New England Journal of Medicine article in 2003, um, that was that clinical trial, but there's been others. And this led to the FDA approval of memantine. So that development of memantine was based on the kinds of work in my lab and others indicating that there's a cytotoxicity involved in Alzheimer's disease. So and I talk about the memantine story in my book, Sculptor and Destroyer, as well as many other interesting stories related to other diseases, Parkinson's, stroke, mental disorders, psychiatric disorders, and so on. Okay, so the accumulation of amyloid beta peptide in the brain cannot be considered a cause of Alzheimer's disease, but it's a contributing factor. Why do I say that? Well, there's many people that live into their 70s, 80s, 90s. They are cognitively sharp. Their brain's working really well, no problem, functioning very well. And then they die from some whatever, stroke, heart attack, cancer. And then at autopsy, neuropathologists have looked at their brains, and some of these people, a significant number of them, have a lot of amyloid beta peptide plaques in their brain, well enough to be considered Alzheimer's disease level. And yet they're okay. Their neurons seem to be working okay. Okay. Now, aging is the major risk factor for Alzheimer's disease. And I've talked about aging in another presentation I've given not too far back on brain bioenergetics. And there's a lot going on in the brain with aging. And there's problems with mitochondria generating enough energy. There's problems in accumulation of damaged molecules in cells, dysfunctional mitochondria, impaired autophagy, uh, reduced antioxidant defenses. And then something we studied a lot, generally with aging, there's impaired adaptive stress responses of neurons, that impaired ability to respond to a metabolic stress and oxidative stress, proteotoxic stress. And so maybe what's happening in these people who have a lot of amyloid in their brain but are cognitively normal is their neurons are able, for reasons we don't know for sure but have some ideas, they're able to resist any toxic effect of the amyloid. Okay. And Parkinson's disease, again, aging's the major risk factor. A lot of changes that are occurring related to aging that are upstream of the pathology that defines the disease. So in the case of Alzheimer's, these changes are upstream of generation of amyloid beta peptide and its neurotoxic effects, and or its neurotoxic effects, or upstream of the tau tangles. And in Parkinson's, these age-related changes are upstream of the alpha-synuclein accumulation in those. And same in stroke again, you have these age-related factors that can precipitate a stroke. Um, Now, I've had Steve Kanan in a couple episodes of Brain Ponderings way back, probably close to 2 years ago, talking about ketones and his work showing that when an individual is in a ketogenic state, either through fasting or ketogenic diet, neurons can switch from using glucose to ketones as their energy source. It's been well known for a long time by PET imaging that people with Alzheimer's disease, the cells have impaired ability to take up and use glucose. We think that at least early in the Alzheimer's disease, well, the neurons are still functional, but not yet degenerated, that they may be able to use ketone as an energy source, even though their ability to use glucose is compromised. And in fact, in a mouse model of transgenic mouse model of Alzheimer's disease that Frank Lafeur's lab developed with our help, we made these so-called 3xTG80 mice that have three different mutated human genes, one amyloid precursor protein, and then tau, and then presenilin 1, which is an enzyme. It's a gamma secretase that liberates the amyloid beta peptide from its precursor. And these mice, over a period of a year, year and a half, up to when they die, about 2 years, they accumulate amyloid plaques, shown here, and intracellular tau, pathological tau. And we've shown that when we put these mice on intermittent fasting or daily time restricted eating with caloric restriction prior to the accumulation of amyloid and the appearance of the tau pathology, and then we let them go for a year, the intermittent fasting can ameliorate their cognitive deficits, as shown here in this maze test. We published that a long time ago, 2007. However, in that study, we found that the intermittent fasting did not reduce the amount of amyloid. So somehow the intermittent fasting was protecting against any contribution of the amyloid to the cognitive impairment. And we also showed that a ketone ester feeding in the diet, these Alzheimer's mice ketone ester can lessen their memory deficits. As I mentioned, some people accumulate a lot of amyloid without cognitive deficits. And there's been this nice neuropathological work staging of the tau pathology shown in the upper panel, immunostaining with the tau antibody, and the amyloid beta peptide plaque pathology shown in bright pink or red in this slide. And so these are called the Brock stage for the tau, Geico Brock, and then Leon-Thal, the amyloid Thal phase. And so there can be people with a lot of amyloid at a level that would be diagnostic of Alzheimer's disease together if they had the tau pathology and still be cognitively normal. However, those people typically have very little tau pathology. So again, similar to these mice in a mice study with intermittent fasting, these individuals are able to resist the tau, which is sort of a you know, indicator of the cells are degenerating. Okay, now I'm going to talk about, I guess, what I'll call the A beta antibody debacle. Now, when I first started working on Alzheimer's disease, doing basic research in around 1990, um, during the first few years, and I was at a really good place, the Center on Aging at the University of Kentucky, Sanders Brown Center on Aging, and Bill Marksberry was the director, world-renowned neuropathologist. And so we were doing basic research in cultured neurons first, and then later in mouse models. And I talked to Bill a lot about the neuropathology, and he saw Alzheimer's patients all the time, and he went to every autopsy personally and helped take out the brain tissue and so on. And and it became clear to me and him as well that the whole answer to Alzheimer's isn't amyloid. And I pointed that out in just briefly in the previous few slides I talked about. But when the amyloid precursor protein gene was cloned and a first family was identified in which a mutation in that gene caused early onset Alzheimer's disease. So John Hardy work and people working with him. And then in this very short but influential article, it's just a hypothesis article and published in Science in 1992, the amyloid cascade hypothesis. John suggested that everything starts with amyloid in Alzheimer's disease, and that's the complete answer to the problem. So if you stop amyloid beta peptide production or somehow remove amyloid from the brain, you will cure someone, prevent or cure Alzheimer's disease. And then Dale Schenk and his colleagues working in a small company found that if they immunize mice, a mouse model of amyloid accumulation, so APP mutant mice that get amyloid in their brain, but I should say no degeneration of neurons in that model, that when they immunize with human amyloid beta peptide, it's also important because the amino acid sequence of human and mouse amyloid beta peptide is different. They found that over time, the amount of amyloid in the brain is reduced. So they had evidence that if they immunize these Alzheimer's mice, the amyloid can be removed by the brain, presumably by activation of the immune system. And then subsequently, they published another paper in mice where they found that if they give monoclonal antibodies to amyloid beta peptide, that can lead to a reduction in the amyloid accumulation in the brain. Now, a big caveat in these studies, in neither study did they look at cognition, learning and memory in the mice, nor did they look at other important indicators of synaptic plasticity. Okay. Okay, so amyloid in the mice can be removed by activating the immune system or with antibodies. And then, we pointed on more than 20 years ago that there's different monoclonal antibodies. They may have different effects. Some may help remove the amyloid, some may not. Some may actually make the toxicity of the amyloid beta peptide worse. So the idea being there that, so if you activate the immune system to break down the aggregated amyloid beta peptide, that you would think at some point during that process, the amyloid beta peptide is going to go back to monomeric and potentially toxic forms of the peptide. And so lo and behold, there's been billions of dollars by multiple drug companies put into, I would say, yeah, a billion overall, at least, into developing monoclonal antibodies, immunotherapies for Alzheimer's disease. All of the clinical trials uh, essentially have been failures, and in many cases, there can be very severe adverse effects of the antibody, in particular brain inflammation, which might make sense. You're activating the immune system in the brain, and also bleeding, microbleeding of small blood vessels, and some people died. Um, and recently, just not too long ago, I think 3 or 4 years ago, the FDA approved one of these monoclonal antibodies for use in Alzheimer's disease, but many, maybe most uh, uh, neurologists are not prescribing them because of the very bad benefit to risk ratio. And there's a lot of scientists now that are really questioning this whole investment in the amyloid cascade hypothesis in terms of funding at the expense of other potential ways of preventing or treating Alzheimer's disease in the early stages. I mentioned the ketones, for example, but you can look at any of those age-related changes upstream of amyloid and as potential targets. Okay. And there's a lot of work looking at the different ways in which these anti-amyloid antibodies can adversely affect neurons and actually promote their degeneration. So there's a nice paper in Nature Reviews Neurology, and this is more than 10 years ago. So this is, in the literature already before FDA approved this monoclonal antibody, and it's really crazy that this was approved. I'm not going to go into the details of, how different antibodies can bind to different regions of the amyloid beta peptide, may have different effects, and so on. Okay. Yeah, so the price of this drug is $56,000 per year, and apparently they were kind of agreed to reduce it to 28,000 per year for this monoclonal antibody that is not going to be helpful. Okay, there have been other approaches that have focused on the amyloid cascade hypothesis, for example, developing drugs that inhibit either base or gamma secretase, the two enzymes that cleave out and liberate the amyloid beta peptide. All of these trials failed, and there's very good reasons why they failed. For example, with gamma secretase, the amyloid precursor protein is not the only substrate that is target of gamma secretase. There's other proteins that are cleaved by gamma secretase. One of them is called Notch, which is a very important signaling pathway in brain development and in neuroplasticity. For example, we showed Yu Wang in my lab and Katsutoshi Furukawa showed through studying Notch antisense transgenic mice that Notch plays a very important role in synaptic plasticity and learning and memory. And then and here's some data from this PNAS paper, National Academy of Sciences paper we published a long time ago. And then we subsequently collaborated with Nick Gaiano when he was at Hopkins and using mice in which Notch has been knocked out only in neurons in the brain that are involved in these synaptic plasticity and learning and memory. And these mice have impaired synaptic plasticity and learning and memory. So the point here is that those drugs, gamma secretase inhibitors that were tried in Alzheimer's patients, they adversely they inhibit notch signaling, and this may contribute to the failures and the worsening of many of the patients in those trials. Yeah. Okay. Um, yeah, and I mentioned the secreted APP alpha. This is pretty interesting because this cleavage, the alpha secretase cleavage, it prevents amyloid beta peptide uh, uh, production. And we showed it has a very important role in controlling excitability of neurons, namely suppressing excitability. So when a synapse is active, this amyloid precursor protein accumulates in the presynaptic terminal. It's released into the synaptic cleft when the neuron fires. And we found in this Nature paper, Katsutoshi Furukawa, now he went back got married to his now Japanese wife and moved back to Japan where he's a neurologist. He's an MD, PhD, still doing a lot of work on Alzheimer's. He found that if you add this secreted APP alpha to neurons, it hyperpolarizes the membrane, so it reduces their excitability. And we found in other studies, this secreted APP can protect neurons against excitotoxicity and against even metabolic insults and amyloid beta peptide toxicity for that matter. And we also showed in this study that the secreted AP alpha can actually have interesting effects on synaptic plasticity. Okay, so that's the A beta amyloid beta cascade hypothesis failure, I guess you'd say. So there's many other strategies. I've talked and we've had podcasts, effects of physical in terms of prevention and risk reduction, or even interventions in early Alzheimer's disease, physical exercise, reducing energy intake, intermittent fasting, uh, cognitive stimulation, keeping your mind intellectually, socially engaged. Um, Again, these have failed, the amyloid stuff. Other things that haven't been tried too much have to do with neurotrophic factors, um, the repurposing of other drugs, for example, certain antidepressants. I think there actually is maybe a clinical trial in early AD of some antidepressants. So there are some other approaches that can be used. Now, briefly, before I go on, I wanted to mention that Alzheimer's disease is defined by the postmortem pathology, certain amount of amyloid plaques, certain amount of tau tangles. But there are other types of dementia characterized histologically. There's some that involve mainly vascular disease. Uh, there's some that are only tau, no amyloid, like frontotemporal dementia. There's something called hippocampal sclerosis of aging, where there's very little amyloid, not much tau necessarily, but massive death of hippocampal neurons. Um, Lewy body disease, which has some alpha-synuclein Parkinson-like pathology, but also dementia, mixed pathologies where you have a mixture of all of these problems. Okay, I'll give you one example, and that's my father, who's Dwayne Matson, shown here in the upper left. So I grew up, actually, I was born in Rochester, Minnesota, went to Mayo High School. Um, my father trained and raced harness horses. This image down here is actually me driving a harness horse. This is probably like the early 1980s. And we had some good horses. We'd race mainly at the county fairs in Minnesota, Wisconsin, Iowa, and sometimes we'd go down to Kentucky. in the fall, entering stake races if we had good race. But anyway, so he was a county attorney for Olmsted County, uh, prosecuting attorney. He actually succeeded a very famous attorney, Frank B. Kellogg. Who was the Olmsted County attorney before my dad, and Kellogg on to become the Secretary of State for the United States. But anyway, so when I was about 6 or 7, my dad bought 100 acres outside of Rochester and had a training track made to train the horses on. So me and my brother and my dad trained the horses, and then we'd go race. And my dad retired early when he was 60 years old because he loved the harness horses, and he could imagine spending the rest of life with them, which he pretty much did for the most part. But then my mom died, and so that was in, let's see, about 1982 or 3, he retired. And then unfortunately my mom passed away in 1992. She had a hemorrhagic stroke, and me and my sister and brother had moved away. So my dad was living alone on the farm with the horses, and he was fine for about at least 10 years or so. And then we started to notice that he had some short-term memory problems. For example, I'd talk to him on the phone, and he'd ask me the same question more than once, which is an indication he forgot he'd already asked me the question. So me and my siblings, we took my dad to the Mayo Clinic and had Ron Peterson, who I knew because he works on Alzheimer's disease. And he was Ronald Reagan's doctor, actually very well known uh, neurologist who focuses on dementia. And so my dad was enrolled in their Alzheimer's research clinical program. So every year he'd go in and do cognitive testing with different tests, many mental state, for example, and and they'd also do MRI of his brain to measure size of hippocampus, for example. And you can see here that we'll just focus on the MMSE. It declines, not necessarily linearly, but just over a 5-year period, there's a very large uh, uh, worsening of his cognition. And on MRI, his hippocampus was getting smaller and smaller during these 5 years, indicating nerve cells are dying. And then finally, well, my brother went to live with my dad, actually, way up here. And then finally, the last couple of years, he was in a special Alzheimer's care place, and we go and see him regularly. Okay, and then he passed away, and then another investigator, a neuropathologist at the Mayo Clinic, Dennis Dixon, looked at my dad's brain under the microscope, and he did not have classic Alzheimer's disease. So his initial diagnosis, as it always should be, is probable Alzheimer's disease. But when they looked at my dad's brain, he had only stage 2 amyloid pathology. So that's, you know, typically that alone wouldn't cause obvious dementia. He had tau tangles, stage 3, so kind of mid-range. He had a lot of TDP-43 pathology, which actually TDP-43 is a protein that accumulates in frontotemporal dementia and also in motor neurons in ALS, Lou Gehrig's disease. Um, he had uh, some vascular changes, not dramatic. Okay, so that's called a mixed dementia. Okay, so anyway, my point there is that um, there's a lot of different changes going on in the brain that are associated with dementia. These are all age-related, and intervening with any one of these may or may not have a clear effect in a patient. Okay, just quickly, a couple other problems with the pharmaceutical industry uh, beyond what I mentioned already. You know this one, the opioid epidemic and overprescribing of opioids and the devastation it's caused and all the lawsuits and and so on. And uh, opioids are a big market, nearly $10 billion a year, and a lot of money has to be spent to treat the problems with addiction. Uh, a lot of people dying from overdoses of getting Narcan out there, so that's a good thing. Okay, so this is an example where opioids were overmarketed, overprescribed, and it's caused a lot of problem. Oftentimes, people get addicted to the drugs because they're given them if they have a surgery, for example, or some other, severe pain syndrome, and then they get addicted, and some of them may then go to the street to get maybe more potent, dangerous opioids. Okay, so I mentioned the idea of reinventing drugs, so developing a drug by different drug companies or even the same drug company that do the same thing, and then marketing one as being better than the other. So that's true with statins for controlling cholesterol, these TNF, tumor necrosis factor, antibodies to suppress inflammation in arthritis. Um, these all these can have really severe effects actually by suppressing the immune system. ACE inhibitors, these angiotensin converting enzyme inhibitors, these are very good for blood pressure, and there's all these different ones that can be chosen from. And none of these have been compared head to head in the same clinical trial. All of these are based on clinical trials comparing the drug to no drug, placebo. Um, and so then uh, drugs that for anxiety, uh, SSRIs, the many different ones, they all do pretty much the same thing. There's a big market for all of these. So a lot of different drug companies making money, spending a lot of money on advertising. If you're really good at advertising, you get one of your drugs to be in the news all the time. Uh, for example, can you say Ozempic in the case of GLP-1 inhibitors when there's actually at least a half a dozen or more uh, of these GLP-1 receptor agonists that do the same thing as Ozempic? So marketing comes. So some of the best treatments for neurological disorders are old and inexpensive. Uh, One example is lithium. Very interesting, right? It's an element. In fact, in the evolution of the universe after the Big Bang, the hot Big Bang, the first atom that arose was hydrogen. The second was helium, which we have in the sun. Very hot up there. Uh, the third is lithium. This three protons and three electrons. And yet this lithium is highly effective in treating bipolar disorders, still the gold standard, very inexpensive. There's still a lot of work developing drugs for bipolar disorder. Interestingly, we don't know for sure how this lithium works. I did some work when I was a postdoc. It was known that it can inhibit calcium channels in neurons, and I mentioned, well, we've talked extensively in multiple podcasts about calcium's role in learning and memory and normal plasticity and disease. Another example, which I'll finish up on, is psychedelics. I've had several podcasts talking about psychedelics. The first one with Franz Vollenweider in Switzerland. Um, was a friend of Albert Hoffman, who first synthesized LSD and psilocybin, the chemical in magic mushrooms that uh, becoming very clear from the clinical trials is a very good antidepressant, anti-anxiety drug. I've talked to Rob Malinka about MDMA. David Nutt talked about how things are out of whack and government regulation of drugs when we have alcohol that is essentially available to all and not regulated very much that, and it causes more deaths than most of these other ones. Opioids are approaching it, but still, if you take in drunk driving and other alcohol, fatty liver disease and so on, alcohol is worse than these other drugs. Psychedelics are non-addictive with no serious side effects, except in very rare cases with few people have developed psychosis. So anyway, here's a nice article by Robin Carhart Harris on the history of research on psychedelics, which began in 1940s with Albert Hoffman, and then clinical trials in depression and addiction during the 1950s, which showed positive effects. And then, unfortunately, the essentially uh, well, making illegal psychedelics by the US government, which halted research for many decades until recently. And now there's been many recent trials, which I mentioned. There's over 200 clinical trials listed on clinicaltrials.gov in a range of disorders, not only depression, anxiety, PTSD, addictions, but chronic pain, which is often associated with anxiety and depression, OCD, even Parkinson's disease, interestingly, anorexia, autism. Okay, so what about the pharmaceutical industry? There's now many companies, startup companies, and even bigger companies that are have drug discovery and development programs. So their idea now is they're going to take psilocybin, for example, and they're going to tweak the molecule a little and try to make it better. Where actually, probably what they'll end up doing is maybe having a drug that is maybe as good, if they're lucky, as psilocybin, but not better. And the way the system is set up now, these may never be compared head to head. There's some good things going on with the psychedelics. One is decriminalization in certain states. Oregon, for example, I understand Washington, D.C., being decriminalized. Mushrooms have been decriminalized. Manufacturing and synthesis, it would be good to have pure psilocybin, LSD and stuff for clinical use. And then you can get the dose right and not have like a bad trip or other side effects. And then, you know, these clinical trials are done, that's kind of you're in a clinical setting with someone there who uh, knows about what to expect in terms of the person's response and experience with the psychedelics and kind of help them through it. And that's all really good. Um, GLP-1, its normal function is it suppresses appetite. It's released into the blood when you eat, particularly in response to carbs. And it goes to your brain, the hypothalamus, and it affects circuits that control your appetite. So it suppresses appetite. It can also promote glucose uptake by muscle and liver cells. So that is good, can increase insulin sensitivity. But many of the beneficial effects for patients with type 2 diabetes or obesity are due to suppression of appetite. So it's essentially causing calorie restriction, and that's why these are having beneficial effects. So you can get the same effect by reducing your food intake, and then you don't have to pay, or your insurance company doesn't have to pay for the high cost of these drugs, which can be quite expensive. There, as I mentioned, based on this peptide GLP-1, this exenatide, which we worked with a little at NIH together with Josephine Egan and Nigel Gregg, and showed it. And not only is it good in suppressing appetite, but it can also be neuroprotective in experimental models, even in cultured neurons, uh, through activation of its receptors. But different companies have made different modifications of GLP-1. The main thing is that this GLP-1, the normal GLP-1 that your body produces, it's normally cleaved by an enzyme called DPP-4. And that cleavage inactivates or renders GLP-1 inactive. And that can occur very quickly after GLP-1 is released into the blood within a few minutes. So all of these modified forms of GLP-1, they prevent or suppress the cleavage of GLP-1. So for example, some of them just involve this amino acid substitution here right next to the cleavage site, with or without other modifications. Um, right. Okay, so these are typically about $1,000 a month. But for the patient, if you have diabetes or severe obesity, that's probably not bad. You suppress your appetite, and maybe eat $1,000 less food a month. Um, okay, so how do we counteract the dark side of the pharmaceutical industry? One that applies to most aspects of healthcare. In the final episode where I talk about problems with the healthcare system, I'll talk about uh, hospitals, physicians, and insurance. But single payer managed healthcare system, such as Medicare for all, we really need this in this country. Um, we need emphasis both in terms of basic research and and um, and everything from medical school on up, in disease risk reduction and prevention, more primary care physicians, fewer specialists. And in theory, if we have more primary care physicians, we'll need less specialists, less diabetologists, less uh, cardiovascular surgeons and so on, because uh, there'll be less of those diseases, less cancers, for example. Okay, ban direct-to-consumer advertisements from drug companies for prescription drugs. Ban lobbying of healthcare providers or government officials by pharmaceutical companies. In clinical trials, require head-to-head comparisons of a new drug with the most effective older drug. This should be done at least one large clinical trial. Seems silly not to do this. It's a waste of a lot of time and effort, I think, and money. And ban participation in clinical trials of investigators who receive money from pharmaceutical companies. Turns out a lot of clinical trials, there are investigators involved in the trial who get money from the drug company. And clinical trial data should be publicly available, right? One problem now is oftentimes the drug companies, they don't just put out the data. And so it's unclear sometimes whether there may be problems early on that don't come out till after the clinical trial. Now, another thing I want to mention is there are inherited diseases where if you inherit a mutated gene from your parent, you have a 100% chance of developing the disease, sometimes at an early age. So there's devastating childhood disorders, uh, something we've been interested in, uh, early onset Alzheimer's disease or Parkinson's disease. For example, shown here, there's mutations shown in red here in amyloid precursor protein that cause Alzheimer's disease. So these people, if they have these mutations, they have a 100% chance when they're in their 50s of becoming symptomatic. Presenilin 1, which is the gamma secretase that cleaves the amyloid precursor protein to liberate amyloid beta peptide. These mutations are even more uh, consequential in terms of the age of onset. In some mutations, the person, when they're in their 30s, late 30s, they develop dementia. For example, down in Colombia, there's these families that there's kind of inbreeding, and they so you've got a kid in their teens who has a parent who's, say, in their late 30s, who's getting dementia. It's very terrible. These, what we call autosomal dominant inherited diseases, whether it's for a cancer, for a cardiovascular problem, metabolic disorder, certain other brain disorders, they could be eliminated in one generation. And I've talked about this in this publication, which I'd be happy to send you if you're interested. But uh, all you have to do is say when someone's a teenager, with their permission and permission of the parent, you get them genotype, see if they have the mutation. If they don't, that's great. They're not going to get the disease at an early age. If they do, they can, and they're going to get married, or they can choose not to have kids or adopt a kid. So that will ensure that they don't pass their mutant gene on to the next generation. Or they can do what's called in vitro fertilization and embryo selection. So in vitro fertilization, essentially you get the egg from the mom and the sperm from the dad, and you fertilize the egg in a dish, and then the embryo, the cell will start to divide. And then when it gets to, like, I don't know, 8, 16, 32 cell stage, you can implant it in a uterus. But what you can do here is in very early stage, like 2 or 4 cells, you can genotype the embryo at that stage and know whether it has the mutated gene. And then you can select an embryo that doesn't, and then implant that in the prospective mom's uterus. So then you will have uh, all the father's other genes except for the mutated one. Well, another one's from him, and then all the mom's genes, good genes, uh, and so that child will grow up with no chance of developing Alzheimer's at an early age. Okay, so this is my last slide going back to here. So I've talked about pharmaceutical industry today. Next time I'll talk about hospitals, physicians, and health insurance, and maybe sum it up a little more in terms of um, solutions. But a lot of it starts with us being educated, um, interacting with our representatives, local, state, federal, and being careful about media, social media, what you follow and listen to, not believing everything you hear from the media. And then um, also talking to your physician uh, and talking to them about primary prevention and how they can help you work to reduce your risk so that you don't have to take drugs or get surgeries or so on. Okay, thanks. So month of January, we're going to pick up with our regular format with me and guests beginning next week. See you then. Goodbye.